The scope of the presented method is to obtain single and pure glands from fresh plant tissue for molecular analysis. This is accomplished by first harvesting a pitcher from the Nepenthes plant. The tissue is then directly fixed onto a microsampling platform on ice to keep the tissue fresh during the microsampling procedure. Glands are isolated in a precise manner and transferred to the PCR tube before performing quantitative PCR analysis on the harvested material. Ultimately, mechanical microsampling technology is used to obtain single and pure glands from Nepenthes plants in order to quickly and microinvasively provide specific tissue for further molecular analysis.
Carnivorous plants are specialists among the flora because they are able to digest animal protein. Our knowledge how that works on a molecular level is still limited. A good model to address this question is the pitcher plant of the genus Nepenthes, because it has distinct organs, pitchers, which produces glands which contribute to these digestion processes.
In Nepenthes, these glands are located inside the pitchers at the bottom. For molecular analysis, it's necessary to isolate these broad and flat structures having a diameter of about 100 to 200 micrometers directly from fresh tissue. To make it more difficult, the glands are covered by a protective hood having a structure similar to a carport, necessitating three-dimensional observations during sampling, as well as a tool that can move in 3 dimensions in a delicate manner. We learned that current microdissection techniques were not made to solve our problems. A laser, for example, failed because of the high water content of the plant tissue. Therefore, we decided to use and employ a new mechanical microdissection technique called Aureka.